Next, coming to the fundamentals of field level and the laboratory sampling along with the importance of statistical tools in food analysis and testing. In this, first of all, we need to understand what is sampling. It is a process of selecting a small portion of sample from the largest lot in order to represent the entire lot. It is the critical step in the food analysis to ensure the results are accurate, consistent and reliable. If we compare the multiple small portions, then we get the accurate, consistent and the reliable answer. And what are the types of sampling? There are two types of samplings, field level sampling and the laboratory sampling. And the field level sampling involves collecting the food samples directly from their natural or the production environment, that is from farms, storage facilities or production lines. The purpose is to assess contamination, nutritional levels or the overall quality at the source. Next, coming to the laboratory sampling, it is a process of preparing and analyzing the collected samples under controlled conditions in a lab. It performs precise and detailed tests to identify contaminants, nutrients and the microbial presence. What is the importance of sampling? It ensures representativeness of the sample, it reduces testing costs while maintaining accuracy and helps to identify the contamination or quality issues without testing the entire population. Population means the sample which we are going to test. Ensuring representativeness of sample means it involves selecting a sample that accurately reflects the population characteristics, means the sample characteristics. This can be achieved by using random sampling and sufficiently large sample sizes to minimize the variability. Coming to the fundamentals of field level sampling, it is the initial step where food samples are collected from their source. These sources can include the farms, production facilities, warehouses or the retail stores. What are the key principles? In this we see the representative sampling, means the sample should represent the entire population or batch. For example, if analyzing wheat from a large truckload, samples should be collected from multiple points. That is the representative sampling. Next, avoid contamination. Use sterile equipments and proper techniques to prevent introducing external contaminants into the sample. Sample size. The amount of the sample collected should be enough to conduct all planned tests. What are the methods of field sampling? Random sampling means select random points to collect the sample. An example is picking random oranges from the truckload for pesticide testing. Stratified sampling. Divide the lot into smaller sections. Smaller sections means strata. Lot is the collection of items and the sample from each. An example is sampling milk from the multiple sections of a tanker to account for stratification. Systematic sampling. Collection of samples at regular intervals or locations. Example, taking one sample every 100 kg of the grains. What is the importance of field sampling? It identifies contamination or the quality issues at the source and reduces the need for extensive testing at later stages and helps track issues back to their origin. And what are the fundamentals of the laboratory sampling? Once the samples arrives at the lab, they undergo preparations and testing. This ensures the sample is suitable for accurate and the repeatable analysis. The key principles are the sample integrity. It maintains the original properties of the sample during the storage and the preparation. Homogenization. Mix the sample thoroughly to ensure the uniform distribution of the components. Next is the use of control. It includes the known standards or controls for the reliable comparisons. This means these standards serves as a baseline to measure and evaluate the results reliably. And the steps in the laboratory sampling, receiving and labeling. Assign unique identifiers to samples to prevent mix-ups. And the storage, use appropriate storage condition, that is refrigeration for perishables, means which undergo degradation or deterioration. Next is the preparation. Example is grinding wheat for moisture analysis or filtering liquid samples for microbial testing. Next, fourth step is testing. Use chemical, biological or the physical methods to analyze the sampling. Next one is statistical tools in the food analysis. Statistics helps to validate the results of the food samples and testing. and it ensures that the conclusions are reliable and scientifically sound. Key applications for statistical tools: sample size determination. Statistical formulas, example power analysis, calculate the number of samples required for reliable results. Common analytical errors, that are instrumental errors, means faulty or uncalibrated equipment. Next is the operator errors, mistakes during the sample preparation or the testing. Next is the environmental contamination, that is introducing external factors like dust or the microbes into the sample. Some practical examples are: here are a few real world scenarios to understand sampling and testing better. First one is microbial analysis in milk field sampling, that is collect milk samples from different points in a storage tank and dilute the sample and inoculate it on nutrient medium. Next, testing process uses statistical tools to determine the average bacterial count. By practicing the statistical methods, we can understand the bacterial count in the milk content. Next one is the pesticide residue testing in the fruit field sampling. Randomly select fruits from the different orchard locations and uses chromatography to detect the pesticide levels and compare results with the legal limits using the hypothesis testing. And some more examples of food testing and the statistical applications, that is microbial testing. The test is the presence of the pathogens like salmonella or the E. coli. For this, the statistical tool used is chi-square test to evaluate the significance of the contamination level across the different batches. Next is nutritional analysis. Test is to measure of protein, fat or the vitamin levels. The statistical tool that is used is regression analysis to correlate nutrient levels with the storage time. And the chemical contamination, it is used to detect the pesticides or the heavy metals. The used tool is ANOVA to compare contamination levels in the different regions. The summary is: importance of statistical tools helps to reduce the variability and improve the reliability and ensure compliance with the regulatory standards and optimize quality control process. And by integrating sampling fundamentals and the statistical tools, food safety and the quality standards can be maintained effectively. Field level sampling focuses on obtaining a sample that represents the entire populations while avoiding contamination. And laboratory sampling involves preparing and analyzing the sample in detail under controlled condition to obtain precise result. Here we should focus on the controlled conditions so that we get the precise results. Both processes are interconnected and critical to overall quality and the reliability of the food analysis. The fundamentals of the field level and the laboratory sampling includes the following critical elements. Here typical objective includes that the quality control, that means the food products meets the quality standard, and the safety analysis, that is detecting the contaminants like pesticides, heavy metals or the pathogens, and the regulatory compliance which ensures the food products meet the legal and the regulatory requirements. Next is the ensuring the representativeness of the samples, means a sample should accurately reflect the characteristics of the entire lot of the batch. Ensuring the representatives involves the proper selection techniques, that is random or stratified sampling or adequate sample size to minimize the variability in the samples, and using appropriate techniques and the tools. And next one is the preserving the samples integrity during the transport and the storage, and preparing and analyzing samples correctly in the lab, and employing statistical tools for the accurate and the reliable results, following strict quality assurance protocols to minimize the errors. These fundamentals are the backbone of the effective food testing, ensuring accuracy and the reliability throughout the process. Now we will see the differences between these both, that is field level sampling and the laboratory sampling, so that we get a proper idea about this topic. First one is definition. The process of collecting the samples directly from the source, that is from farms, warehouses or the production sites. Here the process of preparing and analyzing the collected samples in the controlled laboratory environment. The process is to obtain a representative samples from the entire lot or the population for testing. Here, to process, prepare and analyze the samples for precise measurement and results. Here the environment is the conducted outdoors or in production or storage settings. In laboratory samples, it is conducted in the controlled sterile laboratory settings. Techniques used are the random, systematic or the stratified sampling techniques to ensure the representativeness. Here the sample is homogenized, weighed, diluted and extraction methods for the accurate testing. And the equipment required are the basic tools like the sampling scoops and sterile containers and bags for transportation. Here the testing is done in the fields itself. Here the advanced equipments like the centrifuge, spectrometers and the chromatographs and the microscopes are used in the labs. Next, coming to the sample size, the larger sample sizes are often required to ensure the representation of the lot. Here the smaller prepared portions of the field samples are used for analysis. Focus is to emphasize on the collecting the samples correctly and avoiding contamination during the collection and transport. Emphasis on precision, accuracy and reliability of the test results. Transport and the storage. Samples are transported to the lab requiring proper labeling and preservation. Samples are stored under controlled conditions, that is refrigeration, until analysis. Common errors are the sampling bias and the contamination during collection or insufficient sample size, and errors in the preparation, that is improper homogenization and instrumental issues. Example is collecting soil samples to test for contaminant and selecting grains from a silo, a large storage structure, and the testing the collected soil for heavy metals using atomic absorption spectroscopy. In this diagram, we can see the entire food sampling process starting from the field level sampling, transportation to the laboratory and the laboratory analysis and the data analysis with the statistical tools. This image shows the step-by-step process of the food sampling and the analysis. It highlights field sampling and transportation, laboratory preparation, testing and the data analysis, making it perfect for the educational purpose. Overall, in order to say, statistics improves the precision and credibility of the food analysis processes. Next is data analysis. Uses measures like the mean, median and the standard deviation to summarize and compare the results. Example is analyzing fat content in multiple batches of the milk. Next is the quality control. Control charts track quality metrics. Example, acidity or the microbial count over time. Next is the hypothesis testing. Determine if observed differences are statistically significant. Example is testing if pesticides level in different batches of produce are significantly different. And trend analysis. Use regression analysis to study trends in the quality parameters over time. Example is tracking spoilage rates under different storage conditions. These are the statistical tools in the food analysis. If you like the video, please do like, share and subscribe.